Well, I think, I talked a little bit about this on the panel too. When you're taking such a big idea like this from early stage all the way to, where we're targeting to initiate first in human clinical studies in the second half of next year for our liver-based product. You have to look at is how do you de-risk this over time? So throughout the history of Miromatrix, we took the whole porcine liver matrix, used perfusion decellularization. We commercialized a couple products with that. Got 510K through the FDA. And today they've been transplanted and continue to transplant into thousands of patients. So we really de-risked the matrix itself. And then we published in Nature Biomedical Engineering. We just had another publication on the recellularized liver into an acute liver failure model as well that just came out a couple of weeks ago. First, solving the vascular challenge, demonstrate that you can revascularize the matrix, place that back in and get long-term perfusion of that. And then you can add hepatocytes into that and maintain that level of perfusion, survivability, and start to look at the functional implications of that. So I'd say the journey, as we think about how are we going to bring this next generation technology, life-saving technology together is really, many little steps that continue to build as we solve these challenges to get to the point now that, we're on the doorstep of human clinical studies. So from that standpoint, it's been a fabulous journey. And really what drives us is that patient need, the need for the patients at the end of the day to be able to receive these therapies.